Hello everyone, welcome to another video of molecular basis of inheritance. How are you doing? I hope you are doing good. So in the previous video you're talking about the genetic code. In this video now we will understand the mechanism of translation. So having understood 50% of protein synthesis, now how this information which is copied from the DNA into mRNA now will be translated into protein, into a polypeptide in the cytoplasm. Transcription happens in the nucleus, translation will happen in the cytoplasm of any cell. Okay, so let's begin directly with the topic. So what is translation? Basically it is the conversion of the information which the mRNA brought from the nucleus. Whatever the information was brought in the form of mRNA from the DNA will now be converted into protein. Will now be converted into protein. Okay, just like for making a recipe, you bring about all the all the raw materials, all the ingredients, and then you start making a dish. So now the making of the dish is the second part. First part is preparing all the ingredients, right? You require so and so ingredients in so and so quantity. Okay, then these are the other factors that you need to take care while cooking. So that is, let's say, the transcription. Now you are converting everything into a whole one dish. Okay, something completely different. So similarly here the mRNA will be translated, the information will be translated. Like you have the recipe of the dish, okay, in the form of mRNA. Now this recipe you will convert it into an actual dish. So this conversion, translation of the written recipe into a food, into a cooked food dish, that is translation. Okay, so the mRNA translated and amino acids will be added in a specific sequence to make a protein, that is to make a polypeptide on ribosomes. We have understood the role of ribosomes in one of the videos. If you have still not checked it out, please check it out, wherein we have discussed what is mRNA, what is tRNA, what is rRNA, and their role in translation. Okay, in detail. So you have to see that video first, because here we will just name, okay, tRNA, RNA, and their structure and function is discussed in detail in the previous video on the on the video naming RNA and its types. Okay, so just like transcription and DNA replication, here also the process is divided into three steps: initiation, elongation, and termination. Start, elongation, and stop. Before that, before that, uh, this is the main hero of the translation process. This is the important part or entity which brings about the translation, that is your tRNA. For the process to start, the tRNA has to be charged. For your phone to work, for your phone to do calls or to deliver messages or to do any activity, you require charging of the phone. Similarly, to make the protein, the tRNA has to be charged with an amino acid. Okay, so let's begin. First, we will begin with charging of the tRNA. Okay, let's understand the translation in a gist. So basically, translation is a process of synthesis of polypeptides. Yes, the actual protein synthesis. As you can see, you have the eukaryote, you have the prokaryotic cell, then you have the DNA. From the DNA, you have the mRNA, which is made. Sorry, this is the DNA template. From the DNA template, transcription will happen, mRNA is made. This mRNA will participate in the protein synthesis, that is, it will undergo translation and a protein will be formed. Yeah, this mRNA will be transported from the nucleus to the cytoplasm. So It is a process of synthesis of polypeptides, takes place at the ribosomes, requires several kind of RNA. In translation, you require all the three RNAs. You require mRNA, you require transfer RNA, and this mRNA and transfer RNA will make the protein where on the ribosomal RNA. Translation will take place in the cytoplasm. Okay, so this is a brief understanding of the translation. Let's begin with the first step. That is, first we will understand what are the things that are required for translation. So obviously you require this tRNA. We have discussed the structure in detail in the previous video. So you require tRNA, you require amino acids. As we have just saw, we have seen in the video of genetic code that amino acids will make up the polypeptides. Proteins are made up of what? Amino acids. So 20 different amino acids are composed of proteins. So you require amino acid, you require mRNA, which will bring the information according to the information sequences, according to the code present on the mRNA. That particular amino acid will be assembled and a polypeptide will be formed. So mRNA, which carries the coded information for protein synthesis, then its codons are recognized by tRNA. So amino acid, mRNA, tRNA. Okay, tRNA will bring the required amino acid to the place of protein synthesis. So tRNA will pick up specific amino acid from the amino acid pool and they will carry over the mRNA strand. Okay, fine. So let's say, I'll just give you an example. Let's say this is your mRNA, the sequence is A, U, G, let's say G, G, G. So first three sets is coding for, this is your mRNA. This is coding for methionine. So this tRNA, let's say methionine is here. So this tRNA carrying methionine will be brought over here. Okay. Then the next tRNA carrying, let's say valine, for example, GGP is coding for valine, for example, just assume. So then methionine attached to valine, then another amino acid, then another amino acid, then another amino acid. Like this, the polypeptide will be formed. Okay, so you require amino acid, you require mRNA, you require tRNA. Okay, then also you require ribosomes where this whole assembly will happen. mRNA is there, tRNA is bringing amino acids. Where it will bring? So the factory setup or the place, the site, the place where this whole assembly of the amino acid into a polypeptide chain will take place is at ribosome. So you have the bada subunit and the smaller subunit. The bigger subunit, smaller subunit. Here will be the mRNA, tRNA will come one by one and the protein will be made. So ribosomes are required, ribosomal RNA. Okay, then also you require the binding sites. Like you can see in this picture when you see inside what is happening in the ribosome. So something like this. So the ribosome is having three positions, EPA, we're going to come to that. Large subunit, small subunit, this is your mRNA. This is how the mRNA will position itself and tRNA will come one by one, bringing in the amino acids. Just like RNA polymerase in case of mRNA synthesis, it brings nucleotides and makes the mRNA. In DNA replication, we saw DNA polymerase bringing one nucleotide and making the daughter DNA strand. In the same manner, tRNA will bring one amino acid and the polypeptide will be made. Now, before we begin the process, let's just understand these sites also. In the ribosome, there will be, so the tRNA will come and bind, and it will drop the amino acid. So you have three binding sites, EPA. Let's begin with the P site first. Here, the tRNA attaches to the growing peptide chain. Okay, so like one tRNA bringing one amino acid. So let's say this from the second position, the middle position where it is coming and attaching itself. So this is the peptidyl site, P site. What it is known as peptidyl site or the P site. A site, amino acyl binding site. Amino acyl binding site. where the tRNA carrying the binds the tRNA carrying the next amino acid. Okay, so here is already one amino acid. This is the place where both these amino acids will be bounded, a bond will be formed, and here the another tRNA is ready with another amino acid. Is ready with another amino acid. Once the bond takes place, this there will be a shift, and then this will be bonded to this amino acid at this position. So bond taking takes place at P site. Peptidyl bond forming. So you have one amino acid, another amino acid, another amino acid. So joining, binding takes place at P site. Okay, bringing in. This is the door, let's say, gate. Amino acyl site binds the tRNA carrying the next amino acid. So the tRNA brings and it is ready with the next amino acid at the A site, amino acyl binding site. And E site is the exit site. Here, this is the last amino acid that carried the last amino acid, or you can say the first amino acid. So then this will drop the amino acid. The tRNA here will drop it. I mean, it will bind to the next amino acid on this tRNA, and it will exit site. You will see the animation and you will see in much detail, and then you will understand. Okay, so the PAE sites are there in the larger subunit of the ribosome, which will facilitate the growing of the polypeptide chain or the formation of the polypeptide chain. Now we begin with the first step, that is the charging of the tRNA. The tRNA first has to be loaded with, just give me a minute. Yeah. So first step is the tRNA should be ready with the amino acid. Yes or no? So the tRNA has to be ready with the amino acid. So if you can see over here, the first step is obviously requirement of the energy. This is the enzyme which will cause the tRNA to be loaded with, like you have a truck and the truck has to be loaded with goods for the good to be transported from one place to another. So the truck is here the enzyme. Okay, the goods is your tRNA with amino acids. Okay, then this tRNA with amino acid will be required, will be transported from the pool, from where the charging is taking place to the place where the translation is taking place on the ribosome. Okay, so this enzyme, which is known as amino acyl tRNA synthetase, very important. You have to know the name, amino acyl tRNA synthetase. So a specific amino acid will bind to this enzyme along with the use of energy. This amino acid is there along with the use of the energy. Okay, this is an energy requiring process. Then the amino acid is now activated, bond will be formed. Okay, the bond will be formed, and then the tRNA will come. Now this enzyme will attach this amino acid to that particular tRNA, depending upon what is the sequence. Okay, so the correct tRNA. That is why the word correct tRNA, not any tRNA, not any tRNA will come. Let's say if the sequence is AUG and this is methionine amino acid, then only this tRNA will be attached to this amino acid by this enzyme amino acyl tRNA synthetase. So the correct tRNA binds to the synthetase enzyme. Amino acid is then covalently attached to the tRNA, and then the energy is utilized and the used energy in the form of AMP is released. And now the charged tRNA is released. Now you can see the tRNA is ready with the loaded amino acid, and now it will be released by the enzyme. Now this enzyme will charge the another amino acid with the tRNA, then the another. So this process is cyclic. Okay, it will go on and on. Understood? So amino acids are activated and linked to the tRNA in the presence of this enzyme known as amino acyl tRNA synthetase. Very important. Okay, this is known as the charging of the tRNA. Fine. Now, after the charging, the tRNA is ready. Now the process where the translation will start. Okay, initiation complex will be formed. What are the things that will be required for the start to happen? Charged tRNA, smaller subunit, and the mRNA, right? You have the mRNA strand, smaller subunit, that is a 30S ribosomal subunit, because in eukaryotes, the smaller subunit is of the 30S type. And then this is the first tRNA loaded with the first amino acid, that is methionine. Now this is F-methionine. This is picture is depicting translation in prokaryotes. So UAC is the sequence. Clear? So if this is AUG, the start codon, so the complementary sequence, A pairs with U pairs with A, and G pairs with C. So the complementary sequence UAC will be present on the anticodon binding site of the tRNA. So all these all these things will assemble and will make an initiation complex. Another molecules like a group of initiation factors, these are also small factors, like helping hands, like group of proteins will assist in setting up the assembly, in assembling the ribosome around the mRNA. So this ribosome will be properly set around the mRNA. Okay. All these proteins, these initiation factors will come for a shorter period of time. Once they come, they assemble, they will do their work, and then they will go. Okay. So the initiation factors temporarily recognize specific sequences in the mRNA. They will come and they will locate where the initiation has to start, where the translation has to start. They will bring all these initiation complex participants over here, and then they will recognize, assemble, and then they will go. Fine. This is how the setup will look like. A small ribosomal subunit will bind to the mRNA, initiate the tRNA. This is the first tRNA which will begin the process. That is why it is known as initiator tRNA. Anticodon and codon. If AUG is the start codon, what will be the complementary sequence present on the tRNA? UAC. This is the mRNA. This is the smaller subunit. Okay. So the small subunit will recognize, then the larger subunit will come and sit on top of it. Okay, so this is how the first setup will look like. The smaller subunit, mRNA, then the initiation codon, on that the initiator tRNA carrying the first amino acid, that is methionine. All this is ready. Now bigger subunit will come and sit on top of it. Okay, like this you can see now. So the start codon, the near the beginning of the mRNA is a codon called the start codon. It is usually the methionine. And now we saw the three regions, right? The P site, the E site, and the A site. Now you can have a look over here. Each site will fit a single tRNA. You can see over here. So the process begins here. The first will be here. Now this, can you see the another tRNA getting ready, is ready with another amino acid proline, CCG on the mRNA. So what will be the complementary sequence? C pairs with G, G, and G pairs with C. So now this is coming and this will come and sit over here. Okay. And then this shift will move. There will be a bond formation between methionine and proline that we will see. Understood? So like this. Now the bigger, when the P site is filled, the bigger subunit, that is the larger subunit, will come and sit. This is the final initiation complex, or the start process, uh, you know, uh, starting process, how the setup will look like. This is the starting point. Initiator tRNA occupies the P site. In initiation, the assembly of the ribosome occurs with the AUG start codon within the P site. Smaller subunit, mRNA with the start codon, with the anticodon, the correct tRNA with the loaded amino acid, that is the charged amino acid, and then this A site is ready to bring the next amino acid. Clear? This is how the initial setup will look like. The same picture. You can have a clear look at this. Okay. This we have just mentioned that tRNA whose anticodon is complements the codon on the mRNA and it enters the A site. Now CCG will bring proleu. Okay, so that correct amino acid will bring, and by enzymatic reaction, now the amino acid between P and A will join together. So P and A, proline will come over here. There will be joining of both the amino acid by a peptide bond. And we are all aware, you should be knowing what is the bond that exists between amino acids in a protein, peptide bond. Okay. So as the peptide bond is formed, the amino acid is released from the tRNA in the P site. Okay. So when this comes over here, both of them bonds. So this amino acid is released from this tRNA. Okay, this is over here. Then proline will come over here. A bond is created. This bond will break. This bond will break. This tRNA is now free. It will move to E site, exit site. Now this bond will break. This is the exit site. Okay. So the ribosome then moves one codon down the mRNA, and then basically this moves, or this whole setup moves in this direction. This whole setup will move one codon over here. Now it will move towards here. The ribosome then moves one codon down the mRNA in this direction. Okay. So like this you can see in the picture. Can you see? This tRNA will once the tRNA makes a bond between the incoming amino acid and the existing amino acid, it leaves. So the tRNA will leave, and because the whole setup shifts over here. So like this, one by one amino acids are added. Simple to understand. So this is how the elongation will take place. Okay, tRNA in the P side enters to the E side and leaves the ribosome. So this tRNA, once bond is created, creates the bond between the two amino acids, it moves to the exit side and then it leaves. Okay, and then the A side keeps on bringing another amino acid till it reaches the stop site, and then it moves to the P side. A new mRNA codon is then exposed on the A side, then tRNA with an anticodon complementary to the exposed mRNA codon then enters the A site, and then the process repeats itself. Okay, so first in the P site amino acid, then in the A site another amino acid. Both the amino acid will come together, will make a bond. Then this tRNA is now free. It will leave the P side and move to the E side. This whole setup shifts one by one, and the P and A constantly are engaged in making peptide bonds between the existing polypeptide chain and the amino acid that is brought in one by one, depending upon the sequence which is there. Okay, so this is how the elongation will take place and stop when the ribosome and when the setup encounters the stop codon UGA. Now no amino acid is over here, so there will be release factors which will come into picture. So termination will begin once the end of the mRNA is reached. Proteins called release factors will now come in the A site. No amino acid here, now release factors. So they do not carry any amino acids. These release factors will not carry any amino acid. Okay, so the process of translation is stopped at this point. Fine. So once the stop codon is here, release factor will come. They will not bring any amino acid. So this is the final polypeptide chain which is formed, and then the process of translation will stop. Okay. Once the process of translation is stopped, what happens? All the assembly will detach itself. As you can see over here, as you can see over here, the small subunit, the bigger subunit, the mRNA, the tRNA, and the polypeptide chain, everything is uh, released. So the release factor hydrolyzes the bond between the last tRNA at the P site and the polypeptide, releasing them, and the ribosomal subunit dissociates. So everything will be disassembled, and then this polypeptide is free now, and it will fold and make a protein. mRNA will degenerate, tRNA will be recycled again and again, and these rRNA are there in the cell will be recycled again and again. That is the ribosomal RNA. So have a look over here. This is how the transcription took place. mRNA getting formed. One by one, the nucleotides are coming in. Then this mRNA moves to the cytoplasm. Now you can see one by one the amino acid is coming, peptide word formation, then that tRNA goes like this. Okay, now you can have a better understanding of how one by one the amino acids are brought in, and the tRNA, once the bond is formed, it releases. Ribosome detaches, mRNA moves, and this is how the polypeptide is there. It will roll accordingly, and then the protein is formed. This is a summary of the whole process. You can have a look, pause the video and have a look, go through it, read it again. Yes, step by step. This is another summary, detailed summary. You can see step number 1, DNA in the nucleus serves as the template. mRNA is made, then mRNA is traveled now. Okay, now mRNA moves to the cytoplasm, becomes associated with the ribosome. Then the tRNA will come charged with the amino acid, and one by one, as you can see, the peptide is polypeptide is getting formed. Yeah. Then the charged tRNA with the amino acid will bring attached to the existing polypeptide, leave. Another will come attached to the existing polypeptide, leave. So like this, the whole process will take place. That's all for this video. In the next video, we will talk about gene expression and regulation and lactose operon. Till then, keep revising. Take care. See you in the next video.